This protocol is easy to perform. The cloning phase allows a larger number of T clones to be obtained from fresh samples. Moreover, the phenotype and function of T cells can easily be analyzed. Demonstrating the procedure will be Mary Christopher, a doctorate student from my laboratory.
To induce T-cell cloning, use the bag spike with the needle-free valve to open an irradiated Buffy coat bag in a laminar flow hood. Transfer up to 50 milliliters of blood from the bag into a T150 flask and add 70 milliliters of PBS to the flask. Carefully layer 30 milliliters of the diluted blood onto 20 milliliters of density gradient in each of 4 50 milliliter conical tubes and separate the cells by density gradient centrifugation. Carefully aspirate the supernatant and transfer the PBMC from each tube into a single new 50 milliliter tube. Use PBS to bring the final volume to 50 milliliters and collect the PBMC by centrifugation. After a second wash under the same conditions, suspend the isolated feeder cells in 10 milliliters of cell culture medium for counting and add 25 milliliters of a 70 milliliter cell culture medium supplemented with 50 units of IL-2 solution into each of two 50 milliliter tubes. Then add feeder cells to one tube at a 2 by 10 to the 6 cells per milliliter of cell culture medium supplemented with IL-2 concentration. To set up a T lymphocyte culture, pool the T cell clone-containing supernatants from stenotic valve sample flasks into a single